Before the elegant double helix of DNA became the blueprint of life, before complex proteins built the machinery of every cell, there was a void, a chaotic, primordial world simmering with potential. How did the intricate dance of life begin from such simple chemistry? The answer may lie not with the master blueprint, but with its forgotten ancestor, a single, versatile molecule that could hold a secret and build a world. This is the story of RNA, the rebel molecule that could store information like DNA and catalyze reactions like a protein. It was the jack of all trades at the dawn of time, a chemical spark that may have solved biology's greatest chicken and egg paradox. We journey back 4 billion years to a planet unlike our own to uncover the story of the RNA world. A time when a single type of molecule reigned supreme, writing the first chapter in the epic tale of life on Earth, a story whose echoes are still found within every one of our cells today. In the grand theater of life, every living cell follows a precise and ancient script. This script, the blueprint for everything from a bacterium to a blue whale, is written in the elegant language of DNA. Its iconic double helix contains the instructions, the genetic code that dictates the form and function of every organism. But a code is useless without a reader. The work of building, maintaining, and operating the cell is carried out by an entirely different class of molecules, proteins. These are the microscopic machines, the catalysts and structural components that translate the DNA's instructions into action. They build the cell walls, digest food, carry oxygen, and replicate the DNA itself. Herein lies one of the most profound paradoxes in all of science, a classic chicken and egg problem at the very foundation of existence. To build a protein, the cell must read the instructions from DNA. But to read the DNA and replicate it for the next generation, the cell needs proteins. One cannot exist without the other. So, which came first? How could such an intricate, interdependent system ever have gotten started on a barren, primordial Earth? It is a puzzle that has perplexed thinkers for generations. A system so complex seems to defy a gradual, stepwise origin. It appears irreducibly complex, as if it must have sprung into being all at once, a notion that runs contrary to everything we understand about natural processes. For life to begin, there must have been a way to break this cycle of dependency. There must have been a simpler starting point, a precursor system that did not rely on this dual molecular monarchy of DNA and protein. Scientists began to search for a candidate, a single entity that could bridge this seemingly impossible gap. They needed to find a molecule that could both store information, like DNA, and perform chemical work like a protein. It was a quest for a ghost in the machine of life, a missing link in the chain of creation that stretched back 4 billion years. The search would lead them down an unexpected path, away from the familiar pillars of biology and toward a more versatile, more ancient, and far more mysterious molecule. The answer to the primordial paradox was not in DNA or protein, but in a world that existed long before them. Imagine a world unrecognizable to us today. 4 billion years ago, the Earth was a violent and chaotic place. Volcanoes scarred the surface, spewing gases that formed a thick, alien atmosphere. Oceans, perhaps green from dissolved iron, churned under the gravitational pull of a moon that was much closer than it is now. There were no continents as we know them, just volcanic islands and protocontinents adrift on a global sea. This was the crucible of life, the setting for the first act of biology. In this tumultuous environment, the elegant and stable structure of DNA seems an unlikely pioneer. The double helix is a masterpiece of information storage, its 2 strands protecting the genetic code from damage. But it is also relatively inert. It is a library, a magnificent archive, but it does not act. It relies entirely on a complex entourage of protein machinery to be read and copied. It is difficult to imagine how this sophisticated system could have assembled itself from the chemical chaos of the early Earth. This realization pushed scientists to consider a radical idea, that the world of DNA and proteins we see today is a later, more advanced stage of life's evolution. Before this, there must have been a simpler age, a biological Stone Age, governed by a different kind of molecule. The focus shifted to a close relative of DNA, a molecule known as ribonucleic acid, or RNA. At first glance, RNA seems like a less impressive version of DNA. It is typically single-stranded, making it less stable and more prone to error. In modern cells, its roles often appear secondary, a temporary messenger carrying instructions from the DNA to the protein-building factories, a component of those factories, or a regulator of gene activity. It was seen as a supporting character, a helpful assistant in the grand drama of the cell. But its very simplicity and versatility held a clue. What if its secondary roles today were echoes of a time when it played the lead? What if this humble molecule was once the master of its own world? The hypothesis began to form. Perhaps life began in an RNA world, a time when RNA and RNA alone managed both the storage of genetic information and the catalytic activity necessary for life to persist and replicate. This was a bold and speculative leap, proposing that life's intricate dual system was preceded by a single molecule show. It was an idea that, if proven, would fundamentally change our understanding of our own origins. The appeal of the RNA world hypothesis rests on the remarkable dual nature of the RNA molecule itself. Unlike the rigid and stable double helix of DNA, RNA is a single strand of nucleotides. This structural difference is not a weakness, but its greatest strength. A single strand is flexible, free to twist and fold back on itself, forming complex and specific three-dimensional shapes. Much like a single piece of string can be tied into an infinite variety of knots, an RNA strand can fold into a myriad of structures, creating intricate pockets, grooves, and active sites. This ability to form complex shapes is the key to its power. It meant that RNA had the potential to do something that DNA could not: act as a catalyst. Proteins perform their work because they fold into precise shapes. An enzyme, for example, has a specifically shaped active site that fits its target molecule perfectly, like a key in a lock, allowing it to accelerate a chemical reaction. Scientists began to realize that RNA, with its own talent for folding, could theoretically do the same thing. It could form its own active sites and catalyze reactions. This was the first half of the solution to the primordial paradox. RNA could potentially be the worker, the enzyme, the machine. The second half of the solution was more straightforward. RNA, like DNA, is a nucleic acid. It is built from a sequence of 4 chemical bases. This sequence can carry information, just as the sequence of letters on this page carries information. It can serve as a template, a blueprint from which copies can be made. Therefore, RNA could also be the information storage system, the genetic script. It was the biological equivalent of a Swiss Army knife, one molecule with 2 critical functions. It could be both the blueprint and the builder. In this hypothetical RNA world, the first self-replicating entities would have been RNA molecules. An RNA strand could carry the instructions for its own replication, and its folded shape would provide the catalytic activity to actually carry out that replication, creating copies of itself from the smaller chemical building blocks available in the primordial soup. This elegant concept solved the chicken and egg problem in a single stroke. It provided a plausible, step-by-step pathway for life to begin. But for years, it remained a compelling but unproven idea. The central question lingered. Was there any actual proof that RNA could truly act as a biological catalyst? Proposing an RNA world was one thing, explaining its origins was another. Even if RNA could solve the paradox of replication, a fundamental question remained. Where did the first RNA molecules come from? RNA is a polymer, a long chain made of repeating units called ribonucleotides. Each ribonucleotide is itself a complex structure composed of a sugar, a phosphate group, and one of 4 nitrogenous bases. How could these intricate building blocks have formed on a lifeless planet from simple, non-biological chemicals? This is the challenge of prebiotic chemistry. The quest to answer it began in 1952 with the famous Miller-Urey experiment. Stanley Miller and Harold Urey simulated the conditions thought to exist on the early Earth, sending electrical sparks, representing lightning, through a mixture of gases like methane, ammonia, and hydrogen. Within a week, they found that complex organic molecules, including several amino acids, the building blocks of proteins, had formed spontaneously. This was a landmark result, proving that the transition from simple chemistry to the building blocks of biology was not just possible, but perhaps inevitable. However, creating the building blocks of RNA proved to be a far greater challenge. For decades, chemists struggled to find plausible pathways. Trying to synthesize ribonucleotides in simulated primordial conditions often led to a sticky, useless tar or a mixture of countless unwanted byproducts. The components, the sugar, the phosphate, and the base, were difficult to form and even more difficult to join together in the correct way. The problem seemed so intractable that some scientists began to doubt the entire RNA world hypothesis. But in 2009, a team led by chemist John Sutherland at the University of Cambridge announced a major breakthrough. They demonstrated a plausible pathway in which the building blocks of ribonucleotides could self-assemble under conditions likely present on the early Earth. Their process bypassed the difficult step of joining a pre-made sugar and base. Instead, they showed how the entire nucleotide could be built up from simpler precursor chemicals in a sequence of reactions powered by ultraviolet light and phosphate minerals, perhaps occurring in shallow, evaporating pools on a volcanic landscape. This work, and subsequent research, revitalized the field. It showed that the bricks for the RNA world were not impossibly complex. They could have been forged in the geothermal and photochemical furnaces of the young Earth, accumulating in protected environments, waiting for the next crucial step, the moment they began to link together and form the first functional polymers. For much of the 20th century, the RNA world remained a tantalizing but purely theoretical concept. The central dogma of molecular biology was clear. DNA stores information, and proteins do the chemical work of catalysis. RNA was just the messenger between them. The idea that RNA could be an enzyme was considered biological heresy. That all changed in the early 1980s, thanks to the parallel work of 2 independent research groups, one led by Thomas Seck at the University of Colorado Boulder, and the other by Sidney Altman at Yale University. Seck was studying how RNA is processed in a single-celled pond organism called tetrahymena. He was trying to identify the protein enzyme responsible for cutting a piece out of an RNA molecule and splicing the remaining ends together. To his astonishment, after meticulously purifying his samples, he found that the RNA was capable of cutting and splicing itself, with no protein assistance whatsoever. The RNA molecule was its own enzyme. He and his team were initially skeptical, running every possible control to rule out contamination by a hidden protein. But the result was undeniable. The RNA was the catalyst. At the same time, Sidney Altman was investigating an enzyme called RNASEP, which is involved in processing transfer RNA molecules in bacteria. His group discovered that the enzyme was a complex of both protein and RNA. The conventional wisdom was that the protein part performed the catalytic work. However, through a series of elegant experiments, Altman and his student, Lawrence Goulian, demonstrated that the RNA component alone was sufficient for the catalytic activity. The protein subunit merely helped, but the RNA was the true functional core. These two discoveries, published separately, sent shockwaves through the scientific community. They were the first concrete examples of what came to be called ribozymes, RNA enzymes. It was the smoking gun that the proponents of the RNA World hypothesis had been searching for. Here was definitive proof that a nucleic acid could perform the functions once thought to be the exclusive domain of proteins. The theoretical possibility had become an observed reality. For their revolutionary work, Thomas Seck and Sidney Altman were jointly awarded the Nobel Prize in Chemistry in 1989. Their discovery transformed the RNA world from a speculative hypothesis into a leading and robust theory for the origin of life, opening up entirely new avenues of research into the dawn of biology. In the chaotic theater of early Earth, a cosmic lottery was underway. Countless molecular fragments, forged in hydrothermal vents or delivered by comets, churned in the planet's warm, shallow waters. Among them were the building blocks of RNA, nucleotides. For life to begin, one of these chance creations had to do something unprecedented. It had to make a copy of itself. This is the defining act of life, the spark of heredity, and it represents one of the most profound mysteries in science. The journey to the first replicator was not one of conscious design, but of chemical inevitability. An RNA strand, by its very nature, is a template. Its sequence of bases, adenine, guanine, cytosine, and uracil, forms a specific pattern. In a solution rich with free-floating nucleotides, these individual units would be attracted to their counterparts on the template strand, A pairing with U and G with C. Slowly, painstakingly, a new, complementary strand would begin to assemble, like letters snapping into place against a stencil. Early laboratory experiments, pioneered by scientists like Leslie Orgel, demonstrated that this non-enzymatic replication was possible. But it was fraught with problems. The process was incredibly slow, taking days or weeks to copy even a short segment. It was also riddled with errors, creating imperfect copies that would degrade the original information. Most critically, it suffered from what is known as the strand inhibition problem. The newly formed RNA copy would cling tightly to its original template, forming a stable double helix and preventing either strand from serving as a template for further rounds of replication. The process would grind to a halt after a single copy. The RNA World theory proposes a stunning solution to this impasse, an RNA molecule that could not only store information, but could also act as an enzyme to catalyze its own replication. This hypothetical molecule is known as an RNA replicase ribozyme. It would be the undisputed monarch of the primordial soup. Such a molecule would function as a true catalyst, grabbing nucleotides from the environment, adding them to a growing chain with speed and accuracy, and finally releasing the finished copy so the process could begin again. In modern laboratories, researchers like Nobel laureate Jack Szostak have used directed evolution to coax RNA molecules into performing feats of catalysis, inching ever closer to creating such a replicase from scratch. The discovery of a self-replicating RNA would be a monumental step in understanding our origins. But even with this power, a lone replicator would be adrift in a vast, indifferent ocean. To truly establish a foothold, this spark of life needed to protect itself, to create a boundary between its internal chemistry and the world outside. It needed a home. The first replicator, a masterpiece of molecular chance, faced an immediate crisis of existence. In the boundless ocean of early Earth, it was profoundly vulnerable. The precious nucleotide building blocks it needed for replication could drift away, diluting into uselessness. The delicate chain of information it represented could be torn apart by harsh ultraviolet radiation or disruptive chemicals. For the story of life to continue, the replicator needed a sanctuary. It needed a container. The solution did not require a new biological miracle, but rather a simple consequence of physics. The answer lay in a class of molecules known as lipids. These molecules, which would have been abundant on the early Earth, possess a curious dual nature. They have a head that is hydrophilic, meaning it is attracted to water, and a tail that is hydrophobic, meaning it is repelled by water. When scattered in water, these amphiphilic molecules perform a remarkable act of spontaneous self-organization. To hide their water-fearing tails from the surrounding liquid, they automatically arrange themselves into hollow spheres called vesicles. The result is a lipid bilayer, a stable membrane that forms a distinct internal compartment, separate from the external environment. This wasn't life, but it was the architecture of life. Evidence suggests these essential lipids were readily available. They could have been synthesized in the complex chemical environments near undersea hydrothermal vents. They may also have arrived from space. Analysis of meteorites, like the famous Murchison meteorite that landed in Australia in 1969, revealed the presence of a variety of organic compounds, including fatty acids capable of forming stable vesicles in water. The cosmos, it seems, was seeding our planet with the very ingredients for cellularity. These primordial membranes were not the complex protein-studded barriers of modern cells. They were simpler, more permeable structures. This leakiness was a critical feature. It would allow small molecules, like the nucleotides needed for RNA replication, to diffuse inside from the surrounding water. At the same time, the membrane was strong enough to trap the much larger RNA replicator within. For the first time, a fundamental division was made: inside versus outside. A private chemical world was born, a protected space where the concentration of life's ingredients could increase, making reactions more efficient. This simple vesicle was more than a passive container. It was an active participant in the origin of life, creating the arena for the next great act. The stage was set, the sanctuary was built. Now, what would happen when the replicator took up residence inside? When the first RNA replicator found itself encapsulated within a lipid vesicle, something entirely new came into being: the protocell. This was not yet a living cell in the modern sense, but it was a revolutionary step beyond a mere collection of chemicals. It was an integrated system, a partnership between information and containment, where the whole was far greater than the sum of its parts. Inside its protective membrane, the RNA replicator could now work with unprecedented efficiency. Nucleotides, diffusing in from the outside world, were now trapped and concentrated, providing a ready supply of raw material. The replicator could now build copies of itself much faster than it ever could in the open ocean. This success, however, created a new set of challenges. As the RNA molecules inside multiplied, they took up space and increased the internal concentration of the protocell. This caused water to be drawn in through the membrane via osmosis, making the entire vesicle swell. The protocell was growing, but it was also becoming unstable. It needed a way to expand its membrane and, ultimately, to divide. The solution was elegantly simple. The growing protocell could incorporate free-floating lipids from the environment directly into its own membrane, allowing it to expand without bursting. Division was likely a crude, physical process. Stretched to their limits, these growing spheres might have been split into two smaller daughter protocells by the force of waves, by passing through small pores in rocks, or simply by the inherent instability of their shape. The crucial outcome was that each daughter protocell could inherit some of the precious RNA replicators from the parent. This was the dawn of cellular heredity. For the first time, natural selection had something more substantial to work with than just naked molecules. It could now act upon a population of competing protocells. A protocell containing an RNA replicase that was slightly faster or more accurate would reproduce more rapidly. Its lineage would flourish, passing on its superior genetic information to more and more descendants. Protocells with slower or error-prone replicators would be outcompeted, their unique information lines fading into obscurity. This was evolution in action, a powerful engine for innovation, driving these simple entities toward greater complexity. Yet, for all its success, the protocell was still fundamentally limited. Its RNA could do one thing exceptionally well: make more RNA. To build a more sophisticated and robust machine, to truly master its environment, it needed a new class of molecules, powerful, versatile tools that could perform a vast array of functions. The RNA world was about to invent its greatest creation: protein. The protocell, a triumph of replication and containment, had reached a functional plateau. Its world was built entirely of RNA and lipids. To advance, to build structures, to catalyze a wider range of reactions, and to create a more resilient form of life, it needed proteins. Proteins are the true engines of the biological world, molecular machines of incredible diversity and power. But they are built from a completely different chemical language, the language of amino acids. The monumental challenge facing the RNA world was one of translation. How could the four-letter alphabet of RNA bases be used to specify the sequence of the 20 different amino acids that make up proteins? This is the genetic code, the universal dictionary of life, and its origin is one of the deepest questions in biology. There is no obvious chemical bond that connects a sequence of RNA bases to a specific amino acid. The link appears arbitrary, a convention that had to be established. The solution likely emerged in stages, involving the evolution of new kinds of RNA molecules that acted as intermediaries. The key players in this ancient drama were the ancestors of molecules we know today as transfer RNA or tRNA. Imagine small, foldable RNA molecules, each with a unique shape. By chance, one of these proto-tRNAs might have developed a pocket on its surface that had a slight chemical affinity for a particular amino acid, for instance, glycine. Another might have developed an affinity for alanine. Over time, this weak association would be refined by selection. At another location on this proto-tRNA molecule was a sequence of 3 RNA bases, what we now call an anticodon. This sequence could bind to a complementary 3-base codon on a longer messenger RNA strand. This was the breakthrough. The tRNA became the adapter, the physical link between the language of nucleic acids and the language of amino acids. A primitive ribosome, likely made entirely of catalytic RNA, would have been the final piece of the puzzle. This molecular machine would facilitate the process, holding the messenger RNA template in place and helping the tRNA adapters find their correct positions. As the tRNAs lined up in the order specified by the messenger RNA, the amino acids they carried would be brought side by side, allowing the protoribosome to forge the peptide bonds between them, linking them into a chain. A protein was born. With this incredible innovation, the RNA world had cracked its own code. It had developed a system for manufacturing bespoke molecular tools based on the instructions stored in its genetic library. The implications were staggering. The protocell was no longer limited to the catalytic abilities of RNA alone. It was on the verge of unleashing a chemical revolution that would change the planet forever. The invention of translation was the tipping point. The first proteins, synthesized by an RNA machine from an RNA blueprint, began to populate the protocell. This marked a fundamental power shift in the story of life. While RNA was a versatile jack-of-all-trades, proteins were masters of their craft. Composed of 20 different amino acid building blocks, proteins could fold into an almost infinite variety of complex three-dimensional shapes. This structural diversity made them far superior catalysts. A protein enzyme could perform a chemical reaction thousands or even millions of times faster than a ribozyme. A powerful feedback loop was now in motion. A protocell that, by chance, possessed an RNA sequence that coded for a useful protein would gain an enormous competitive advantage. Perhaps this first protein was an enzyme that helped repair the cell's membrane, or one that improved the synthesis of nucleotides. The most valuable protein of all, however, would have been one that helped replicate RNA itself. A protein polymerase, working alongside the original replicase ribozyme, would have supercharged the process of reproduction. This new era is often called the RNA protein world. RNA still held the ultimate authority as the keeper of genetic information, but it began to delegate most of the day-to-day labor to its powerful protein creations. The ribosome, the cellular factory that builds proteins, stands today as a magnificent molecular fossil from this transformative period. It is a large, complex machine composed primarily of RNA, a clear relic of a time when RNA managed the world. But even this successful partnership had a vulnerability. RNA, for all its talents, is not an ideal molecule for permanent information storage. The sugar in its backbone makes it chemically fragile and prone to breaking down. As genetic archives grew longer and more complex, this instability became a critical liability. The final step in establishing the modern biological system was the transition to a more durable medium, DNA. The process likely involved a protein enzyme, itself born of the RNA world, that evolved the ability to perform reverse transcription, reading an RNA template and synthesizing a DNA copy. DNA offered two huge advantages. Its double helix structure is far more stable than RNA's single strand, protecting the precious genetic code from damage. Furthermore, its two-stranded nature provides a built-in backup. If one strand is damaged, the cell can use the other as a template to perform perfect repairs. With this last innovation, the modern architecture of life fell into place. DNA assumed the role of the secure, long-term genetic archive. RNA became the versatile middle manager and worker, the messenger, the adapter, and the structural core of the factory. And proteins became the primary workforce, carrying out the vast majority of cellular functions. The RNA world had effectively made itself obsolete, handing over its dual roles of information and catalysis to more specialized molecules. The reign of the single molecule was over, but its legacy was immortal, forming the foundation of every living thing to follow. The RNA world was a whirlwind of creation and collapse. Its single-stranded, chemically reactive nature made it a brilliant catalyst for life's first steps, but also its greatest weakness. Every copy was an approximation, every reaction a gamble against molecular decay. For life to evolve beyond simple, self-replicating loops, it needed a foundation that could withstand the test of time, a library that would not crumble. Out of this primordial chemical soup, a new molecule began to emerge. It was a subtle variation, a molecular cousin to RNA, yet its design was a revolution in stability. This new contender was deoxyribonucleic acid, or DNA. Its innovation was twofold and profoundly elegant. First, it swapped RNA's sugar, ribose, for a slightly different version called deoxyribose. The removal of a single oxygen atom from the sugar backbone might seem trivial, but it rendered the entire molecule far less reactive and resistant to breaking down. It was like forging a chain from steel instead of iron. The second change was in one of its 4 informational letters. RNA used uracil, a base that could, through natural chemical decay, morph into another base, cytosine, corrupting the genetic message. DNA replaced uracil with thymine, a more robust and distinct letter. This change made it much easier for cellular machinery to spot and repair damage, ensuring the message remained true. But the most profound innovation was its structure. Instead of a lone, exposed strand, DNA formed a partnership, twisting into the iconic double helix. 2 strands, bound together, protecting the precious genetic code within. Each strand was a mirror image of the other, a perfect backup copy. This was no longer a fleeting message scrawled on a fragile scroll. This was an archive built for the ages. The arrival of DNA was not a sudden conquest, but a gradual shift in the balance of power. It offered something the chaotic RNA world desperately needed: permanence. Life now had a master blueprint, a stable, reliable repository of information from which it could build, generation after generation. The frantic pace of RNA's reign was giving way to a more measured, more secure form of existence. The question was no longer about simple survival, but about how to build complexity. And this new master molecule held the key. But how did this newcomer, a molecule that could only store information and not act on it, manage to usurp the all-powerful RNA? In the grand evolutionary contest for genetic supremacy, DNA possessed a set of overwhelming advantages that sealed its victory. While RNA was a jack of all trades, it was a master of none. Its very versatility was its undoing. DNA, on the other hand, specialized. It relinquished the role of a chemical catalyst and focused on one task: perfect information storage. This specialization allowed it to become exceptionally good at its job. The double helix structure was its ultimate weapon. With 2 complementary strands, the cell had an ingenious, built-in proofreading system. If one strand was damaged by radiation or chemical attack, repair enzymes could read the opposite strand and restore the original sequence with incredible accuracy. This process, known as mismatch repair, reduced the rate of mutation by a factor of thousands. RNA, with its single, exposed strand, had no such safety net. An error in an RNA genome was often permanent and passed on to all descendants, leading to a high error catastrophe rate where accumulating mutations would eventually render the molecule useless. DNA's stability allowed for the storage of vastly more information. RNA molecules are limited in length. They become too fragile and prone to folding into useless shapes as they get longer. DNA, with its rigid, predictable helical structure, could be millions of times longer. This was the difference between a single page of instructions and an entire encyclopedia. Life could now store the blueprints for more complex proteins, more intricate metabolic pathways, and eventually, multicellular organisms. The division of labor that DNA's rise permitted was also a critical advantage. DNA remained protected within a central compartment, the future nucleus, like a priceless original manuscript in a secure vault. To get work done, the cell would make temporary disposable copies in the form of RNA. These RNA messengers would carry the instructions to the cellular machinery. If an RNA copy was damaged, it was of little consequence. It could be easily discarded and replaced. The master DNA blueprint remained pristine and unharmed. This separation of storage and function created a robust and efficient system. DNA was not just a better molecule, it enabled a better system for life. It provided the fidelity, capacity, and security necessary for evolution to move beyond the microbial mat and begin its long, slow march toward the staggering complexity we see today. The throne was won not in a violent battle, but through sheer chemical and logical superiority. With DNA firmly in charge of the genetic library, what became of the former ruler? When DNA ascended to become the primary keeper of genetic information, RNA was not cast out into oblivion. Instead, it was demoted and repurposed, its former kingdom integrated into the new cellular order. Today, our cells are living museums, filled with molecular relics that bear witness to RNA's ancient reign. These are not dusty artifacts behind glass, but active, essential components of life's machinery. The most stunning of these living fossils is the ribosome. This is the protein factory of the cell, the molecular machine that translates the genetic code into the proteins that perform nearly every task in our bodies. For decades, scientists assumed that the proteins within the ribosome were doing the catalytic work. But in a Nobel Prize-winning discovery, it was revealed that the heart of the ribosome, the very part that forges the bonds between amino acids, is made entirely of RNA. The ribosome is, in fact, a giant ribozyme. This is the ultimate proof of the RNA World hypothesis. The machine that builds the modern cellular workforce is a direct descendant of the ancient RNA toolkit. It's as if we discovered that the engine in a modern supercar was secretly powered by steam. Another relic is transfer RNA, or tRNA. These small, cloverleaf-shaped molecules are the interpreters, the essential link between the language of nucleic acids and the language of proteins. Each tRNA molecule reads a three-letter word in the genetic code and fetches the corresponding amino acid, delivering it to the ribosome. Without these RNA adapters, DNA's instructions would be meaningless. They are the Rosetta Stone of the cell, a fundamental component that almost certainly dates back to a time before DNA and complex proteins existed. Beyond these major players, the cell is teeming with other small RNA molecules that act as regulators, switches, and timers. They fine-tune gene expression, guide modifications to other RNA molecules, and defend the cell against invading viruses. These non-coding RNAs represent a vast, hidden layer of control, a shadow government run by the descendants of the old ruler. They demonstrate that RNA never truly relinquished all of its power. It ceded the throne of heredity to the more stable DNA, but it retained control over the day-to-day operations of the kingdom. Looking at these molecular fossils, we can piece together a picture of what once was, a world where RNA did everything. And the most famous of its retained roles is the one that connects the old world to the new, the role of the messenger. In the sophisticated cellular democracy governed by DNA, information is power. But that power is useless if it remains locked away. The genius of the modern cell is its system for distributing information safely and efficiently, and the key to that system is a specialized form of RNA. This is messenger RNA, or mRNA, the tireless courier that bridges the gap between the protected genetic archive and the bustling factory floor of the cell. The process begins in the nucleus, the cell's command center, where the DNA chromosomes are housed. Here, an enzyme complex glides along a specific gene, transcribing its sequence not into another DNA molecule, but into a complementary strand of mRNA. This act of transcription is like a librarian carefully photocopying a single page from a rare, priceless book. The original book never leaves the safety of the library, but its information can now be transported. This newly minted mRNA molecule is a transient, working copy. It is a single-stranded, disposable message designed for a short but crucial life. Before it can leave the nucleus, it undergoes processing. Sections are spliced out, a protective cap is added to one end, and a stabilizing tail to the other. It is prepared for its journey into the cell's cytoplasm. Once it passes through the nuclear pores, the mRNA's message is delivered to a ribosome. Here, the process of translation begins. The ribosome moves along the mRNA strand, reading its sequence of bases in three-letter words called codons. For each codon, a specific tRNA molecule brings the corresponding amino acid, and the ribosome links them together into a growing protein chain. The mRNA molecule acts as the template, the blueprint that dictates the exact sequence of the final product. This elegant division of labor is a cornerstone of all complex life. It allows for exquisite control over gene expression. The cell can produce thousands of copies of mRNA from a single gene if a lot of a particular protein is needed, or just a few if a little will suffice. When the protein is no longer required, enzymes quickly degrade the mRNA messages, halting production. This ensures the cell doesn't waste energy and can respond rapidly to changing conditions. The mRNA system is a testament to evolution's brilliance, a perfect compromise that leverages the strengths of both DNA and RNA. It uses DNA's stability for long-term storage and RNA's versatility for short-term, high-volume production. But as we look deeper, we find that RNA's role as a simple messenger is just the beginning of a much more complex story. For many years, the story of RNA seemed to end with its role as a humble messenger and structural component. It was seen as a relic, an intermediary molecule that had been overshadowed by the elegance of DNA and the power of proteins. But in the late 20th century, a scientific revolution began, revealing that RNA's kingdom had not fallen. It had merely become a hidden empire, subtly influencing nearly every aspect of cellular life. The first major revelation was the discovery of RNA interference, or RNAi. Scientists found that tiny, double-stranded RNA molecules could effectively silence specific genes. These microRNAs act as the cell's internal police force, seeking out and destroying messenger RNA molecules with matching sequences. This discovery unveiled a previously unknown layer of genetic regulation. RNA wasn't just carrying messages, it was actively deciding which messages were read and which were silenced. It was like discovering that the postal carriers were also in charge of censoring the mail. This opened the floodgates. Researchers began finding a whole universe of non-coding RNAs with diverse and critical functions. They act as molecular scaffolds, guiding proteins to their proper locations. They function as switches, changing their shape in response to environmental signals to turn genes on or off. The more we look, the more we find that RNA is at the heart of the cell's most complex regulatory networks. It is the software that runs on the hardware of DNA. This new understanding has catapulted RNA to the forefront of modern medicine. The development of mRNA vaccines was a direct application of our knowledge of RNA's role as a messenger. By introducing a synthetic mRNA blueprint, we can teach our cells to build a viral protein and mount an immune response without ever being exposed to the virus itself. Beyond vaccines, the potential is immense. Therapies based on RNA interference aim to silence the genes responsible for genetic disorders or cancers. Scientists are designing RNA-based drugs that can target disease pathways with incredible specificity. We are learning to speak RNA's language, to write our own messages and commands into the cell's ancient operating system. The RNA world never truly ended. It evolved. The molecule that sparked life from non-living matter, that once reigned supreme as both gene and enzyme, is still with us. It endures not as a forgotten monarch, but as a powerful and sophisticated force, a hidden kingdom still shaping the destiny of all life on Earth. The quest to understand life's origins is not confined to our own planet. It extends into the silent, vast expanse of the cosmos. As our robotic emissaries venture to other worlds, they carry with them a profound question. Are we alone? And if we are not, might the life we find bear the telltale signature of an RNA world? Astrobiologists now consider this a tantalizing possibility. On Mars, rovers like Perseverance drill into ancient lakebeds, searching not for fossils of complex creatures, but for subtle chemical clues called biosignatures. These could be specific organic molecules that are difficult to produce without biology. A complex RNA molecule, preserved in the cold, dry Martian soil, would be a revolutionary discovery. It would suggest that the chemical pathway to life is not unique to Earth. The search extends even further, to the icy moons of the outer solar system. Jupiter's moon Europa and Saturn's moon Enceladus are believed to harbor vast liquid water oceans beneath their frozen shells. Plumes of water vapor erupt from Enceladus, giving us a direct sample of its hidden sea. Missions are being designed to fly through these plumes, analyzing their composition for the building blocks of life, and perhaps for RNA itself. Finding life that stopped at the RNA stage would be an extraordinary scientific prize. It would be like discovering a living fossil of life's first chapter, a window into a stage that on Earth was completely overwritten by the rise of DNA and proteins. The challenges are immense. RNA is a fragile molecule, easily broken down by radiation and chemical reactions over billions of years. Detecting it would require instruments of unprecedented sensitivity. Yet, the search continues, driven by the understanding that if life could begin here, through the elegant simplicity of RNA, it could happen elsewhere. Every radio signal analyzed, every soil sample vaporized, and every icy plume traversed is part of this grand search, a search for a cosmic echo of our own deepest past. They exist on the very edge of what we define as life. They are not cells. They cannot reproduce on their own. Yet, they are masters of replication, hijacking living machinery with breathtaking efficiency. They are viruses, and within their simple structures may lie a profound clue about the ancient RNA world. A vast number of viruses, from the common cold to influenza, are RNA viruses. Their entire genetic blueprint is encoded in this single-stranded molecule, not DNA. This has led to a compelling hypothesis, that viruses are not products of cellular evolution, but perhaps relics of it, ancient holdovers from a time before cells, as we know them, existed. According to the virus-first hypothesis, viruses may have originated in the primordial soup as self-replicating RNA strands that developed a protective protein coat. They were the original genetic parasites, predating the first cells. In this view, cells evolved later, in part as a defense against these primordial replicators. The behavior of a virus mirrors the theoretical dynamics of the RNA world. An RNA virus is essentially a message, a piece of information, that seeks out a system capable of reading and copying it. This is precisely what scientists believe the first ribozymes did. They sought out raw materials in their environment to create copies of themselves. Viruses simply took the next step, using the complex, pre-built machinery of a cell as their environment. Even more compelling are viroids, the simplest known infectious agents. They are nothing more than a short, naked loop of RNA. They have no protein coat. They are pure, replicating information. To many scientists, a viroid looks exactly like what a successful molecule from the RNA world might have been. Are these tiny agents the descendants of the planet's first replicators? Are they echoes of a 4-billion-year-old biological reality, still playing out their ancient game of survival inside our own cells? The question remains open, but it reframes our perspective on these ubiquitous entities, suggesting they are not just agents of disease, but perhaps living messages from life's distant dawn. For decades, RNA was seen as little more than a cellular messenger, a humble assistant to the master molecule, DNA. But as our understanding grew, so did our ambition. Scientists began to realize that RNA's ancient power, its ability to both carry information and act as a catalyst, could be harnessed. This wasn't just about understanding the past, it was about building the future. The revolution began in the lab. Researchers learned to design and synthesize custom ribozymes, RNA molecules, built not by evolution, but by human ingenuity. These designer enzymes could be engineered to cut other RNA molecules at specific locations, acting like molecular scissors. This opened up a new frontier in biotechnology, offering ways to target and disrupt the genetic instructions of pathogens or faulty genes. Then came a discovery that would shake the foundations of genetics. In 1998, American scientists Andrew Fire and Craig Mello uncovered a natural process within cells called RNA interference, or RNAi. They found that introducing a small, double-stranded piece of RNA could effectively silence a specific gene, preventing its message from being translated into a protein. It was a cellular defense mechanism, likely evolved to fight RNA viruses, but for science, it was a magic switch. Suddenly, researchers had a precise and powerful tool to turn genes off at will, allowing them to decipher the function of thousands of genes in organisms, from plants to animals. The discovery, for which Fire and Mello won the Nobel Prize in 2006, had immediate practical applications. In agriculture, RNAi became a way to engineer crops with built-in resistance to pests, silencing essential genes in the insects that tried to eat them. It offered a greener alternative to chemical pesticides. The ancient molecule was being repurposed, its ability to seek and bind to a specific sequence transformed from a tool of primordial replication into a high-tech instrument for research and industry, setting the stage for an even greater revolution just over the horizon. The promise of RNA as a therapeutic agent simmered in laboratories for years, a concept rich with potential but fraught with technical hurdles. The molecule was notoriously unstable, and the human immune system often attacked it as an invader. For decades, the idea of injecting RNA into a person seemed like a distant dream. But a handful of persistent scientists, like Catalin Carico and Drew Weissman, refused to give up. Through methodical research, they discovered a way to make a crucial chemical modification to messenger RNA, or mRNA, that allowed it to fly under the immune system's radar while still delivering its genetic message. This was the key. Their breakthrough, initially met with skepticism, laid the groundwork for a complete transformation of medicine. Then, a global crisis provided the ultimate test. The sudden appearance of a new, rapidly spreading virus presented an unprecedented challenge. Traditional vaccine development took years, a timeline the world could not afford. This was mRNA's moment. Because mRNA technology is essentially a platform, a delivery system for any genetic code, researchers could move at astonishing speed. Once the virus's genetic sequence was known, they could synthesize an mRNA vaccine, encoding a piece of it in a matter of days. The principle was elegantly simple. The mRNA would instruct the body's own cells to produce a harmless piece of the virus, training the immune system to recognize and fight the real thing without ever being exposed to it. The result was the fastest vaccine development in human history. It was a monumental triumph of science, a testament to decades of foundational research. But this was only the beginning. The success of mRNA vaccines blew the doors wide open for a new class of therapeutics, Today, RNA-based treatments are being developed for a vast range of conditions, from personalized cancer vaccines that teach the immune system to hunt tumors, to therapies that can correct rare genetic disorders by providing a functional copy of a faulty gene. The humble messenger has become a powerful medicine, transforming a primordial biological process into one of our most advanced tools to promote human health. We began our journey in a warm little pond, 4 billion years in the past, with a simple molecule learning the magic of replication. We have traveled through the rise of DNA, the complexities of the cell, and the dawn of human consciousness. Now, we stand at a remarkable threshold. The very molecule that sparked life is now a tool in our hands, and its future is a story we are only just beginning to write. The potential is breathtaking. We are moving beyond treating symptoms and into an era of programmable medicine. Imagine a future where an injection of tailored RNA can instruct damaged heart cells to regenerate after a heart attack, or command the immune system to eradicate a tumor with surgical precision. Consider therapies that can deliver instructions to correct a genetic defect not just for one person, but for their descendants. Technologies like CRISPR, which uses a guide RNA to edit the DNA of a living cell, are a powerful extension of this new paradigm. We are learning to speak life's original language. This newfound power, however, comes with profound ethical responsibilities. As we gain the ability to rewrite the code of life, we must confront difficult questions. Where do we draw the line between therapy and enhancement? How do we ensure these revolutionary treatments are accessible to all, not just a privileged few? The conversations we have today will shape the world of tomorrow. The story of RNA is the story of life itself. It is a story of information striving to persist, of simplicity giving rise to complexity, and of a deep, unbroken chain connecting the first self-copying strand to the scientists who now wield its power in a laboratory. From the primordial catalyst to the programmable cure, RNA has been the silent, ever-present thread. Its past gave us our existence. Its future may well define our destiny. The primal molecule's greatest chapter is yet to be written.